And then Tuesday, we'll talk about the electron transport chain. And then we'll talk about the water in your urine. Very exciting. Oh, what I do for work, it's amazing. All right, so always keeping the big picture. When we talk about cell resp and we're slinging all this vocabulary, I always try to remind you to step back and see the big picture, all right? What are we doing? We're trying to synthesize ATP. We're trying to make the energy molecule so that we stay alive, because ATP really is the energy molecule of life. If you have ATP, the cell will survive. No ATP, cell dies. That's simple, okay? So the production of ATP is kind of important, and all cells do it. Bacteria do cell resp also. They just do it a little bit differently because of course, bacteria are prokaryotes, so therefore they don't have a mitochondrion. Fine. So cellular respiration, 3 steps, okay? Glycolysis out in the cytoplasm, Krebs in the center of the matrix, or the center of the mitochondrion, called the matrix, and then the ETC, which is actually embedded on the inner membrane of the mitochondrion. We'll talk about this on Monday. So step 1, step 2, step 3. The whole point is to make this ATP here, right? So there's energy down here. Where does this energy come from? It comes from all of the high energy electrons that you're ripping out here. So those high energy electrons have to be carried to the electron transport chain. Okay, so everything is about oxidation and then delivery to the ETC. Oxidize the substrate, deliver to the ETC. Take the energy, make ATP, and now you've got these low energy electrons. Get these things out of here. How do you do that? Oxygen and protons. Okay, always step back and get the big picture. So, while this diagram is nice, the official formal Campbell's diagram looks like this. Same concept, step 1, step 2, step 3. The only difference is we added the link reaction in here. Most people group the link reaction with Krebs, therefore we say 1, really step 2 and step 3, but some people split this out to 4. Fine. Again, see all of the electron carriers. Why? Because these macromolecules are loaded with high energy electrons. That's why they're food. Okay. Then you have decarboxylation during link and decarboxylation during Krebs. This is why all organisms release CO2. We breathe out CO2. Fungus releases CO2. Plants release CO2. Protozoans release CO2. Okay. But don't be confused, this is not the CO2 that contributes to global warming, because then this CO2 cycles back into photosynthesis, and that's what we call the carbon cycle. Okay, so don't think, oh, there's more people on Earth, so therefore we're all breathing out more CO2. It's not the way it works, it's the carbon cycle, all right? You breathing out CO2 does not contribute more CO2. It's same CO2 cycling. It's the CO2 we dig out of the ground. All right, so then we zoomed in on glycolysis. Glycolysis, again, big picture idea. Glucose, pyruvate, some high energy electrons, and some ATP, which you discovered is 2 NADH, 2 pyruvate, and 2 ATP. So the net products of glycolysis are all 2s, right? Technically, glycolysis produces 4 ATP, but you have to invest 2, so 2 in, 4 out. That means a net gain of 2. Right? You do produce some ATP. There's no electron transport chain. There's no chemiosmosis. There's no proton gradient at all. This is called substrate level phosphorylation. The enzymes literally just attach the phosphate directly to the substrate, and that's how they synthesize ATP. You, of course, have oxidation and reduction. You have a lot of this in cell rest because you're oxidizing the food that you ate. Plants are oxidizing the G3P that they produced. Okay, this is all about oxidation. Cell resp is an oxidative process. Cool. So glucose in, pyruvate out. Okay, so that's back to here. Glucose in, pyruvate out. All right. So now the pyruvate out in the cytoplasm now needs to enter the matrix of the mitochondrion. Okay, so this reaction here links glycolysis in the cytoplasm with Krebs in the matrix. Thus, we call it the link reaction. So this membrane right here of the mitochondrion, the outer mitochondrial membrane, is the same membrane here. Please be comfortable where you are in the cell. Okay, so this is now pyruvate to a compound called acetyl-CoA. Fine. 3 carbons to only 2. Well, if you go 3 to 2, it has to be 3 minus 1 is 2. Well, if you are subtracting or extracting or pulling out a CO2, that's decarboxylation. So the link reaction is an oxidative decarboxylizing reaction. Oh, there's a mouthful. What does that mean? It means that your substrate, pyruvate, is decarboxylized, right? When CO2 leaves, have a nice day. Then this substrate is oxidized. The high energy electrons are pulled out. Those high energy electrons are bonded to the associated proton and NAD. That is reduction, and that forms NADH, which of course ships off to the electron transport chain. You're then left with a 2-carbon compound called an acetyl group. However, this acetyl group, super unstable because it is loaded right there, loaded with high energy electrons. Right? So since it's unstable, you temporarily stabilize it. Temporarily, that's why that bond is a squiggle. That's a chemist's way of writing, this is temporary, right? Temporarily stabilized by this group here. This is called a metabolic cofactor, and we refer to it as coenzyme A. So here's coenzyme A. You don't have to worry about the structure at all. It's not complicated. It's not doing it. You attach the coenzyme A to stabilize that 2-carbon group. Comfortable?
Yes.
Good. All right. So now, if that's link, now we're sitting here with our 2-acetyl-CoA. So now it's time for Krebs. Okay. So that one looks like this. Now, when students first see it, it's like, oh, it's complicated. It's not. It's really not. So let's zoom in on the top. Just because it's unfamiliar doesn't mean it's complicated. So this is Krebs. Okay, the Krebs cycle happens in the core of the mitochondrion, the very center of the mitochondrion, which is called the matrix. Boom, right? Can you imagine all the jokes we heard when the 3 Matrix movies came out? And I'm like, yes, no Mr. Anderson, all this other kind of stuff, whatever. So here's your 2-carbon compound. So which compound is that? With 2 carbons right on top.
Acetyl-CoA.
Yep, this is acetyl-CoA. Acetyl-CoA with, of course, its 2 carbons, right? You only have to learn 2 other names. You're going to learn this one, and you're going to learn this one, okay? Now, when you take this in biochemistry, if you're a bio major, you will then go through, and this is isocitrate, and this fumarate, and alpha-ketoglutarate, and they have all these names, and you just memorize it, and they give you a test, and you give them the information you memorize, and they go, wow, you're smart. And you go, no, I can memorize, okay? But for right now, I want you to learn that one and that one because they're important, all right? This one is called oxaloacetate. Oxaloacetate. Oxaloacetate has 4 carbons. 1, 2, 3, 4. Oh, make it a little bit bigger. Just like that. Oxaloacetate. So you have 4 carbons here. So a 4-carbon compound is bonded to a 2-carbon compound, giving you a, what's 4 plus 2?
6.
6. Look at you guys, your IB. And this compound is called citrate. Some of your professors will refer to this as citric acid because it ionizes in water, and so fine. And some people even call this the citric acid cycle, or TCC. Wow. The citric acid cycle. Let's see, so that would be T. Can you tell what I don't use this? Citric acid cycle, the CAC. Wow. All it is 4 plus 2 is 6. This group here, the stabilizing group is here. The stabilizing group is not here, so where did it go? Right there. It is then shuttled back from the matrix, back out into the cytoplasm to bond to another acetyl group, which is why then people account for that shuttle, and thus they subtract from the total of 36, dropping it down to 34 or 32. That's why professors give you different numbers. Whatever they give you, keep it. Fine. All right, you now have a 6-carbon citrate. So then you have some molecular shuffling. But how many carbons do you have sitting over here?
5.
5?
Oh, 6, yeah.
Still 6 there. Okay. So did you have any decarboxylation at all?
No.
None. And you say, well, then what happens? Oh, if you really want to know, there's some shuffling of water, right? Why? Because this group, in all honesty, this group moves from here to here, right? Do you need to know that now? Nope. But if you're interested, that's what's going on. It's just a shuffle. Then you go from 6 to how many?
5.
5, I agree. So what do you think goes inside of that little arrow, that little circle? It's more of an oval, really. What goes right there? Since we went from 6 to 5. CO2. What do we call the removal of CO2?
Decarboxylation.
Okay. Now, in Krebs, the reactions over here are coupled. Oxidation, reduction, and decarboxylation happen at the same time. We call it coupled, all right? So there's a coupled reaction here, and there's a coupled reaction here. So in this box here is good old NADH. So what goes right above that?
NAD.
Yep, NAD and its associated proton. So what am I going to put on the substrate, and what am I going to put on the NAD? What happens to the substrate?
Oxidized.
Oxidized, oxidation. What happens to your NAD?
Reduced.
Reduced. These are called coupled reactions because they happen back to back in the same step. What are the names of these substrates? In all honesty, who cares? Concepts. What's happening here? Your substrate is being decarboxylized or decarboxylated, and your substrate is being oxidized for its high energy electrons. These high energy electrons are then being shipped or transported to the electron transport chain. All right, so then we come down here with 5 carbons. Your next substrate, how many carbons?
4.
4. What happened?
Decarboxylation.
Decarboxylation. Decarboxylation. NADH, which would be NAD plus H plus. I like that. Ooh, I didn't put my, yeah. Go like that. A whole bunch of pluses over there. All right. Should I insult your intelligence and ask about oxidation reduction?
Sure.
Sure. Hey, so what happens here?
Oxidation and reduction.
You guys are amazing. All right. So these are coupled reactions. It happens together. You oxidize high energy electrons, decarboxylation. Oxidize high energy electrons, decarboxylation. They're coupled. Fine. What do you notice now about the number of carbons down here and the number of carbons up here?
Same.
They're the same. So do we have any decarboxylation over here at all?
No.
Nope. Let me zoom out on that just a little bit. So 4 plus 2 is 6. 6 minus 1 is 5. 5 minus 1 is 4. And now the same number of carbons cycle back. Okay. So this is 4 plus 2, 6, minus 1, back to 4. So these are all 4 carbons over here. There's no decarboxylation over here at all. Don't have to worry about the names of these compounds. You'll memorize them if you're a STEM major. I'm not going to clutter your head with them now. Now, if you want to know this substrate, after it's oxidized and after it's decarboxylated, starts to get a little unstable. So your coenzyme comes back, stabilizes this molecule, and then leaves. Again, it's a metabolic cofactor. Where do you get it? Vitamins. So when you take your vitamins or you eat green leafy vegetables, it helps support the stability and efficiency of your metabolic process. Thus, you are more healthy when you eat a well-balanced diet. God, I sound like a parent, right? But then you've got some fun and games down here, okay? So, let me get rid of that. Actually, that might be about your graduation. Nope. That's all right. That's right. All right, so the starburst, what always goes inside of
ATP.
that? Always ATP. Now, we said that Krebs makes ATP, which it does. And so right here is ADP. Right here is an inorganic phosphate, okay? Now, here's where the fun begins. Because if you were taking the IB exam, IB only holds you accountable for ATP on the bottom. However, you know how there's 4 bases in DNA, and there's adenine, thymine, guanine, cytosine? Yes? Okay. Adenine is what is used in adenosine triphosphate. One of the things we don't teach in high school until this point is, guess what? There's not only ATP, guess what else there is? TTP, there's CTP, and what's the last one? There's GTP. And you go, what? And you go, yeah. There's actually other energy molecules besides just ATP, and they use those same 4 bases, adenine, thymine, guanine, cytosine. Now, by far, like by a huge margin, ATP is the most common energy molecule by far. However, there are times in the biochemistry when other triphosphates are used. Here's one of them, okay? So if you really want to know the technical part of it, well, as technical as we're going to get right here, GDP picks up an inorganic phosphate and becomes GTP. There's then a transfer here, and the phosphate goes from the GTP to an ADP to make ATP, right? Look, all you have to know is that on the bottom of Krebs, it makes an ATP. I simply introduced this here, so those of you that are going to be bio majors know that there's this other energy molecule called GTP. There's a whole nother layer, as always, of details here. Cell biology and biochem is going to blow your mind. It's so cool. All right, so we make some ATP. Now, there's a different electron carrier here that's not NAD. What's the other electron carrier?
FAD.
Yep, totally agree. FADH2 when it has its electrons. So this would be FAD plus your 2 associated protons. This is electron transport. Excuse me. Well, it is an electron transport. So your substrate is oxidized while your carrier is reduced. Again, big picture idea. NADH, NADH, FADH2. Now, if you want to know, a little water comes in. And you say, why? Because this double bond with the carbon and this hydrogen, this double bond is moved, and therefore the OH group is moved up, if you really want to know. But this will be NADH. Therefore, this is NAD plus your associated proton, which means this substrate is oxidized, this carrier is reduced, and you're back to where you started. And the angels sing. Ah. I probably shouldn't enjoy this much, should I? This stuff's so cool, right? So, if you look at your chart, though, this chart here says that Krebs produces how many ATP?
2.
2, right? But how many did we show here?
1.
1. While it's very possible, did Manro make a mistake? No. Why did I only write 1 here, but I wrote 2 here? It's not a mistake. Why is 2 here, but only 1 here? I'm sorry? Oh, interesting idea. Was the ATP in that? Oh man, that is a great answer. It kills me to say no, but that's a great answer. No, okay, sorry. Okay. The how many acetyl-CoA are produced by link?
2.
2. So how many times does Krebs turn per glucose?
2.
Twice. Okay. 1 glucose makes 2 pyruvate, which means Krebs turns twice. All right. So when they ask you about the products of Krebs, you always have to ask them whether it's per glucose or per acetyl-CoA, because it's twice as much. So let's do per acetyl-CoA. And then over here, we'll go per glucose. I love that answer about the ATP is invested. That was an excellent answer. That was a good one. Nicely done. All right. So what are our products per, let's see, there's going to be anything about that. 1, 2, there's 4 of them. 2, 3, 4. What are our products per acetyl-CoA when this turns once?
1 ATP.
1 ATP. There's 1. Done.
2 carbon dioxide.
2 carbon dioxides. I'll agree with that.
3 NADH.
3 NADH. Almost sounded like there was a question mark there.
And 1 FADH2.
FADH2. Done. Per glucose, what do we do to these numbers?
Double them.
Double them. I'll put them in the same order. Boom. So when you take biochemistry in 3 years, it's just going to be a long way away. It'll be here before you know it. And they ask you for the product of Krebs. Always ask them per what? Per acetyl-CoA or per glucose. It makes a big difference. So what'd you do in class today? Oh, we just talked about Krebs. Easy. Okay. Unfamiliar, it's new. Don't be intimidated by it. Look at the carbons. 4, 2, 6, 6, 5, 4 stays the same. Decarboxylation, oxidation reduction, they're coupled, right? Then you got a little substrate level phosphorylation with, holy cow, there's GTP, get a load of that. This is the only place that you will find FADH2, and you're thinking, why? And I tell you, because I don't know why. But if one of your professors ever explains to you why FADH2 is here, will you please come back and educate me? I would love to know that. And then we get one final NAD oxidation reduction event at the top, and you're done. Okay, so notice oxidation, 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 oxidation. These are all of the remaining high energy electrons that are being ripped out of the macromolecules. Don't forget, these are the same electrons that were excited at the end of the thylakoid process, right? From photosystem 1. These are the same electrons from photosystem 1, right? Everything that's in our food, those electrons were excited by plants in photosystem 1. Even if you're eating protein, right? Let's say you're a carnivore and you're digging into, carne asada. It's the only form of red meat. I can't give it up, right? I've really tried to get rid of red meat, but come on, it's carne asada, right? But anyway, when you eat protein, whether it's chicken or fish or pig or cow or whatever protein that you're eating, if it's an animal product and you think, oh, well, this wasn't absolutely. The plant excited those electrons. The electrons then went into an amino acid into the primary consumer, and now you're eating the primary consumer. It just goes through the organism to whoever's next. All right. So I think that's more than enough biochem for a morning. I will see you Tuesday morning. Don't forget about your CSF cords. If you need me to help you, let me know today, tomorrow, or Sunday. And then we'll do the recording for you. I'll find out about your graduation being together, I will confirm that. Come on, that has to happen. And then the live stream question. And then if you could take care of the RSVP survey for those of you that already have, thank you very much. And then I'll see you all Tuesday morning. We'll do the electron transport chain and then break out into groups. Do presentations the last week in June, and that's how we'll kick off your riding off into the sunset the first week in June.